This is an industry partner panel discussion. There'll be short presentations, as short as our panelists can make them, to leave more time for question and answers. And I think that this is actually a piece that we've not talked a lot about. But if one part of long-term sustainability and global collaboration is leveraging funding agencies and philanthropic organizations like Wellcome, like NIH, then the other part of sustainability, the other part of creating real leverage, is effective partnerships, responsible partnerships with industry. From the beginning, we've had industry participants. Jeff and I reached out recently to probably a dozen or more industry participants about how best they could collaborate. And we're delighted to have 3 of those positive responses represented today in the form of Alan Schuldiner from Regeneron, Meg M from GSK, and Phil Febo from Illumina. And I would really welcome you, ask you to give your short presentations, and then hopefully we have time for audience participation. So Alan, over to you.
Thanks so much, Peter. I've unmuted myself, so I presume you can all hear me well. And thanks for the invite to share some of my thoughts about industry collaboration with the IHCC. You know, when I get these email invites to participate in industry panels, my first reaction is that you got the wrong person, in that it's been relatively recently since I moved into industry, and my brain still thinks very much like an academic. So maybe that's a good thing with regard to providing some insights into how we might be able to work better together, because I know what you're thinking. So I'm sure all of my industry panel have a similar slide in terms of what motivates biotech and pharma to be interested in large cohorts, and in particular human genetics of large cohorts. And this has been mentioned prior by my colleague Gonzalo Abacasis, who presented earlier today. We're interested in identifying new drug targets and pathways. We're interested in applying large electronic health record databases to identify new indications for drug targets and our therapeutic programs. We can use these large data sets to de-risk particular drug targets by confirming lack of on-target adverse side effects in drug target loss of function carriers. And of course, we're very interested in the traditional pharmacogenetics to identify genetic and other biomarkers that will allow us to stratify patient populations so that we could identify these populations that will have greater efficacy, and on the other side, fewer adverse side effects. And really, I can tell you at Regeneron, human genetics now drives the agenda. We have sort of turned the drug discovery pipeline on its head. We start with humans and human genetics, and we only then go to preclinical models like mice and cell systems when we have identified through human genetics the likelihood that a drug target will be effective in humans. So now, about 5 years ago, the Regeneron Genetics Center was born, and we are essentially a comprehensive genome center matrixed within a mid-sized biotech company. And we do ultra-high throughput sequencing, in particular exome sequencing, and have really created automated ways in which we can biobank millions of samples. We can perform DNA sequencing and genotyping in literally millions of samples. And to date, we've now sequenced more than 1,000 exomes. And over the next few years, we expect to be sequencing at least 1 million exomes a year for the next several years. And of course, as you heard in great detail already, these volumes of data that are generated require really the cloud and large-scale computer sciences and computer systems in which not only can Regeneron access and analyze the data, but also our collaborators can access and analyze the data as well. So we now have more than 90 collaborations with academic collaborators and healthcare systems from throughout the world. I've mentioned we've sequenced over 1 million exomes, and I think we've learned a lot of lessons about how to collaborate with academic and centers and healthcare systems And the bottom line is that we have already active collaborations with 4 of the larger IHCC cohorts, and we're in active negotiations with at least 10 other of the IHCC cohorts for collaboration in the genetics and exome sequencing space. So the idea of academic industry collaborations, at least for Regeneron as it relates to the IHCC, is already something that's well underway. And so we really do believe in all of the same currency that the IHCC believes in and that our academic collaborators do. Our model is really to advance understandings of human health and disease through collaboration and sharing. We see the work we do in genetics very much in the pre-competitive space. And as you can see from our track record of high-profile publications, and this is just the tip of the iceberg, we walk the walk in that we share our data through publication. My message here is, so let's get on with it. Let's stop discussing processes and barriers and stop drafting guidelines. I'm only kidding. These things are all important, obviously, but I think we just need to get on with this agenda. And I yesterday read in great detail the guidelines for collaboration with industry. I think the document is fully congruent with Regeneron's principles for collaboration. I've suggested some minor wordsmithing to make the principles a little bit more reciprocal and provided some suggestions there. The principles of mutual respect for each other's capabilities, motivations, much of which we have in common, and our currencies are really important. The ideas of fairness and equity, transparency, declaration and monitoring of conflicts of interest, timely and open dissemination of research findings after short exclusivity periods, as well as the intellectual property and freedom to operate principles that are in that document are all very congruent with how Regeneron conducts its business in terms of these collaborations. One thing that I wanted to stress is that industry should not be regarded as a funding agency. Regeneron, and I know my fellow panelists, we're real scientists, and we expect to be fully engaged in the research as collaborators, much more similar to academic collaborators than as we traditionally have thought about academic industry collaborations. And then just to finalize, my remarks in terms of other considerations, as I've mentioned, these 90 collaborations have taught us that one size does not fit all, and there's a range of opportunities and options for collaboration. Everything from fee-for-service, where there would be no or minimal restrictions and exclusivities, all the way to full or major support for a particular project in which the contracts and terms would be more restrictive and require additional exclusivities. We prefer single contracting entities. Of course, local laws, GDPR, other governing bodies, quite important. Federated data sets for Regeneron will be a little bit of a new territory for us, but this is the way the world has moved and the way Regeneron needs to now be thinking about these collaborations. And just to throw out, a particular model that seems to be working pretty well that could be adaptable for IHCC is the Pharma Consortium model that is funding exome sequencing of all 500,000 samples of the UK Biobank. Also recently now additional funding for multi-omics. And we see this as the ability to generate a resource that has mutual benefits for all. So with that, I think I'll conclude my remarks and look forward to discussion to follow.
Many thanks, Alan. And we'll keep this moving right along. Meg, please take it away and from your role at GlaxoSmithKline.
Thank you for inviting us to speak here today, and thanks to Alan Schuldinger for kicking off this particular session. So today, I want to talk a little bit about GSK's interest in supporting the IHCC. I mean, I think for decades we've been doing all we can to squeeze new insights from old drugs, known mechanisms, animal models, and other clues. And we fail so much more often than we succeed because we don't really understand the diseases very well. And I think everyone is quite aware of how developing new drugs is expensive. In 2015, we published a paper that showed that drugs with human genetic evidence were two times more likely to be successful. And as a result of that, GSK has really built a resource organization that's driven by genetics. It sounds similar to what's happening at Regeneron and many other pharma companies as well. We usually begin with a hypothesis-free approach, such as a GWAS, and then layer on variant to gene mapping, target tractability, fit with unmet need, and developability. And we see a partnership with with organizations like the IHCC to be critical. We want to make sure that you're successful in understanding the genetic underpinnings so that GSK can be successful and focus our efforts on figuring out which of those genes are going to make good drug targets. So, here we talk about the various different, we use genetic and patient data from a variety of sources, some of which are part of the IHCC, including our ICHA, including 23andMe, UK Biobank, FinnGen, and the public data. And in addition, we use deep phenotypic information to try to identify, we see great value in using deep phenotypic information to optimize the indications, such as this PheWAS study I illustrated here for IL-33, where we were looking to really optimize which asthma patients would be the best suited and whether or not there were any unanticipated off-target effects. GSK's data strategy has really focused on collaboration with data sources with large numbers characterized for a broad range of traits. And yesterday, as you went over sort of the criteria for being a member of the IHCC, I could see that all of that was really in line with our main efforts. We see that when I look back at our data strategy and our future investments, we're planning to focus on collaborating with biobanks, and we'd love to collaborate with IHCC with something like was recently reviewed with the Alzheimer's disease. We're certainly interested in high disease burden with effects to leverage data that can enable identification of progression to more severe outcomes, and we're really trying to build up our understanding of the use of electronic health records for this important research. Certainly, we're interested in biobanks that have survey and recall capabilities. And I was really excited to hear about the efforts in expanding the diversity, because we're certainly finding that in some diseases, the diversity is not only going to be important for us understanding what we may see in clinical trials, but also to study conditions that are poorly represented in the current data. An example might be hepatitis B. And so we're certainly, in addition to that, we'll be wanting to help promote and fund cross-biobank research. We see the IHCC as really important in addressing two critical issues in biomedical research. The first is the enabling cross-biobank research, which these graphs, these power curves show will be really critical to get to the numbers that will enable the use of genetics in drug discovery for more than just the most common conditions. And finally, to promote that research that will make sure that we can study conditions that are only going to be, only have good numbers in racial groups that are currently not represented. So thank you, Peter, for inviting me today, and happy to be a part of the panel.
Many thanks. We'll move along to Phil, and I think probably a different emphasis from somebody who's very much been in the middle of this world between cohorts, genomics, and less on the drug discovery or the downstream stuff. So Phil, please bring us your perspective from Illumina.
Well, thank you, Peter, and thank you for Jeff and for Terry for inviting me to participate and inviting Illumina to participate in this during a difficult time. And I really appreciate the creative solution and the perseverance to continue on. As Chief Medical Officer at Illumina, I'm charged with helping ensure genomic information becomes part of the standard of care. And indeed, I was drawn to Illumina, and Jeff, who's been a mentor to me since he recruited me to Duke back in 2004, knows that I have a fundamental belief that an individual genome should be a foundational part of their medical record and the basis for risk-based approaches to healthy behavior, disease prevention, and disease treatment. The genome obviously needs to be incorporated into decision support within systems that healthcare providers use to take care of patients and draw upon over time. And we anticipate a system similar to the PACS system for digital radiology images, where reports are in the medical record, but there's a link to full and comprehensive genomic data for those who wish for more information than provided on the report. Of course, for any technology to be incorporated in medicine, there has to be a sufficient clinical utility, the patient and healthcare benefit, to justify the cost. And over the past 17 years, there have been tremendous progress in building evidence for clinical utility of genomic data and decreasing the cost of sequencing compared to when the first human genome was published in 2003. With respect to the value, we see increasing awareness of pharmacogenomics, hereditary risk for diseases from cardiovascular disease to cancer, rare disease, and most recently, polygenic risk scores. On the cost side, the cost of generating a genome has dropped from approximately $3 billion for the first project to under $1,000 and is moving towards $500 with a path to a $100 genome in the not so distant future. However, even for children suspected of having genetic disease, a whole genome or a whole exome analysis is infrequently performed and often they'll be on a diagnostic odyssey for the early years of their lives. And patients with advanced cancer, they get any molecular test approximately 40% of the time, and only 15% get comprehensive genomic testing to inform decisions. Regardless of the genomic information you study, there's significant underutilization throughout the globe. And there are many reasons for this, but in the end, the fundamental challenge is that healthcare providers do not feel the information within an individual genome yet holds sufficient value for them to ask for the information, and in fact, demand the information. And as I consider where we have to go to get to a critical value proposition for those healthcare providers, you have to get to a tipping point where they demand it in order to have the confidence that they're taking the best care of their patients. And that's why the International 1000K Cohort Consortium and other organizations represented today are critical, because it's only through the aggregation of large cohorts, the deep analysis of those cohorts, including, of course, sequencing, but also quality phenotypic data, that will move beyond the identification of some highly penetrant, rare or uncommon genes for unusual diseases, to understand how common variants with modest impact on disease can be aggregated to refine risk for every individual seen in a healthcare clinic. And indeed, the success of these activities also depends on the standards, the work that GEA4GH has done, as well as interested to hear about PhenoX, because phenotypic standards are also really important. And while polygenic risk scores have started to show what's possible, there remain significant challenges in understanding the polygenic risk scores are validated across populations of different ancestry and ethnicity. Ensuring the greater representation of diverse population is critical, like Meg mentioned, and as mentioned by Alan. And again, it is through sequencing and the federation of large cohorts that provides a path within which advanced analytics can really thrive. Machine learning, AI, they can find non-obvious genetic associations that will help an individual understand their personalized highest health risks and take the appropriate preventative measures. This is what's required to map those M2M chains that Rachel Lau from the Broad in her presentation mentioned as a focus for the International Common Disease Allowance. In the era of COVID, we're looking forward to the rapid identification of host genomics associated with differential transmission and virulence of the disease as an intermediate and compelling proof point of the importance of genomics in what is now a common disease, unfortunately. This is exemplified by what Sir Farrar's comments about the work done by Decode on COVID, leveraging their database in Iceland that they already had in their intense screening of the population. At Illumina, we've moved quickly to help shore up resources for sequencing in China, Africa, Southeast Asia, and US. And I think, Jeff, to your point, this will continue to build the infrastructure and the access to technology that, if organized through efforts like the International Consortium, 1000K Consortium, will really be powerful. Importantly, while we've focused on the variants associated with phenotypes, it's also important to leverage those resources to identify variants that have no impact on phenotype. So we can remove those from consideration of the millions of variants in each individual who gets a whole genome. So with that, I want to emphasize that at Illumina, where we remain highly motivated to partner with these groups, as we've done with GA4GH and others, to provide our perspective on ensuring the technology is readily integrated into the efforts to standardize data, facilitate sharing. Well, I've not read the draft of the guidance for collaboration with industry. I look forward to seeing it because it is critical. I've spent most of my career in academics. I now spent the past 7 years in industry. There is clearly a path for very productive collaboration, and certainly Illumina has a history of that and will continue that. And so I'll stop my comments there. And again, thank everyone for participating today.
Thanks, Phil. I did ask everybody, on all attendees, to, if you have a question, put them in chat. But I think one of the thoughts as I listened to all three presentations, obviously this is a coalition of the willing, and you three organizations, individuals, you get it from a collaboration. You understand the need to come as scientists, not as the marketing people, to be equal partners, to be contributors, all those good things that you and your organizations exemplify. But we heard from Ewan earlier, there is often in the consent codes, there's one that says not for use by commercial research. So as you navigate that water, which you do effectively with many organizations, many cohorts, many biobanks, but what about, if we look at Jeff's question, I had the same thought. What about in the low middle income countries? What about outside your home base operation countries? How do you see the special challenges of creating a trust-based relationship with cohort leaders and governments in LMIC environments?
Maybe I can jump in to start.
Start on.
So actually at the Regeneron Genetics Center, I lead a group called the Founder and Special Populations Group. And one of the major motivations to that group is to better understand the allelic architecture of populations from throughout the world, literally phenotype agnostically. And this is because we believe, as the comment in the chat box suggests, that it's just going to be really important, step one, to just understand the allelic architecture of populations from all over the world. So we have been actively seeking collaborators around the world who have actual access to patient populations or population-based samples in which the consent and local laws permit sharing of samples and de-identified data for sequencing. And like all of our collaborations, sequence data that we produce is available pretty much in real time to our collaborators. And we have very generous data sharing policies where that data can be shared with other academic collaborators and ultimately published on as well, used in variant servers so that local physicians might have a feel for what the local allelic architecture is in quote-unquote normal individuals to help inform exomes of undiagnosed disease patients and the like. So I think the bottom line is that consents permitting with appropriate data sharing, we have been reasonably successful in engaging groups from low and middle income countries.
Yeah, and I would just add, I think, the efforts, as Alan's talking about, for, industry or academic centers with incredible resources to partner with centers in some of these underrepresented and developing countries is really important. I think it's also important is creating a path to building infrastructure within country, building the expertise, really using that activity to build in country, in region, scientific expertise and a foundation toward independence. At Illumina, we're really working hard. We have a range of sequencers and people can step through that. we're also trying to address the bioinformatics challenge where, many labs can get comfortable running a sequencer now, but most are still daunted by the bioinformatics, both in just analyzing the specimens, but also in how do they meaningfully participate in these large collaborations. And I think finally, there has to be an eye towards sustainability. These can be started through grant fundings, and obviously the Wellcome has been incredible there. The NIH with their H3 Africa and other activities has been really supportive, and non-government organizations. But how do these centers get to a sustainable path? And this, I've recently had the opportunity, obviously before COVID, to travel a bit and engage with some of these centers. And for instance, Ethiopia or Egypt or Morocco, they have the talent, but getting to that sustainable model where they have resources coming in to support continued genomic research and genomic testing. There's still challenges there, but I certainly think COVID will heighten the understanding of the importance of this, because in-country surveillance is going to be critically important, as was mentioned earlier. And it could very well be a nice path to build upon to go into more non-communicable diseases, sequencing, et cetera, to build from infectious disease surveillance.
Meg?
Yeah, I wanted to point out that, like the portal, that a couple of the things the IHCC is doing that I think is going to make a huge difference for groups like GSK, for companies like GSK, and that is really understanding what is available and what trait information is there. I've actually, as I've like Phil, traveled and visited with many biobanks, what I find to be kind of the most interesting aspect, and then I think something that IHCC is really going to be uniquely positioned, is the gap in understanding about what can be done, what traits, what data, what is there, and as so much as that you can make it on a portal where you can browse it and understand it, and then figure out who do I talk to and what are the policies, and getting to the point where we can start to use some of the cloud-based infrastructure that's being built and leveraging that. I think all those things are going to really democratize the access to data and researchers in a way that can be really daunting. If you don't have the opportunity to travel to so many places or are not really sure what's available. So I think some of those things are going to be really transformational for helping us understand that. And certainly, we see in many respects a GSK or other, we can't replace that local knowledge that you have, the understanding of your record, the understanding of the people. I mean, that's got to be done locally. And I'm hoping that initiatives like this group can help make that easier for researchers who are just getting up to speed.
So Meg, you actually anticipated one of the questions that was coming up, more or less as you were speaking, from Lucia asking, for those not from industry, from those running the cohorts, what are the pain points that industry experiences and that the cohort leaders can help you address? I think this findability, that knowing where the data is clearly one of those pain points. Alan, Phil, anything else that jumps off the page as what would facilitate you being even stronger collaborators?
Well, I guess one of the pain points that we sometimes come across is the breadth of phenotype data that might be shared for any given collaboration. Just as, if we're going to expend major resources to sequence the entire exome or the entire genome for that matter in tens or hundreds of thousands of individuals, we'd like to utilize that data to cast the broadest net possible across the clinical phenome. And this requires, fairly broad agreements for sharing of the phenotype data. And as I mentioned, in this world now of federated data sets, data harmonization, all of the topics that were covered here in the last couple of days, I think we're all broached with challenges as to how that's actually going to work.
Yeah, I think I would just reflect Alan's comments about standardization. Peter, I don't think I have to talk to you about the importance of standardization, both on the molecular phenotypes, the molecular genotypes, and the phenotypes, and collecting that phenotypic data with the right granularity is very challenging. I would also say trust is a big challenge, where large consortium grow comfortable with the best practices with engaging with industry. It's hard for some of these labs to really understand and trust when a big global corporation's coming in to consider collaboration. And that's why setting standards is really important for those collaborations. So people, who do want to collaborate have kind of a guide to help them as a resource.
Again, I'll pick up something from the chat, but I think generally I've seen, we've been trying to make that same point from a IHCC or a GFGH perspective. The idea of industry coming in as a group of collaborators, whether it's the cloud companies or pharmaceutical companies, is an easier thing for a consortium to onboard than feeling that somebody have got to select one supplier or one partner. But the question that came up, Alan, perhaps this was picking up on your point with your UK Biobank example. So the concern is about where data is not either consented for private use, or this idea of setting up a new arrangement. And is it the limited period of exclusivity that industry can support an initiative for a limited exclusivity, knowing that after that, the data is available for all? And we've seen that model used, but perhaps if you just give us a little bit more example of how you see this as a sustainability option.
As I said, we've done now, more than 90 deals with collaborators from around the world. And, while there are some guiding principles, I could say that no two are the same, but some of the guiding principles are. And the idea of some period of exclusivity, seems to be something that everyone can get around, and in which academic collaborators can have immediate access to the data. And, to be honest, some of our academic collaborators want longer periods of exclusivity with their data than the companies actually want. Again, I'm being a little bit facetious here, but it works on both sides. And I think periods of exclusivity are one good way to get around some of these issues. If the consent forms, say that there can't be sharing with other commercial entities, then sort of that's a red flag. We obviously can't proceed unless potentially the collaborators can get reconsent from a subset of individuals who would be willing to enter into collaborations with private entities.
Yeah, and I think you just anticipated. The next question was asking about exclusivity when you've got multiple industry partners. So you've got both sides of this coin, the need to form coalition of industry supporters, strategic partners, but on the flip side, what's your incentive? So I think, let me just check time. I think we're probably reaching the point at the end of this session, and we'll move into the overall wrap up, which Terry is going to lead for us. But just before we do, open mic back to all three panelists. Any one closing thought you'd like to leave us with in the next minute and a half?
Thank you for inviting us. I think it's, I know over the last 2 days, it was just really exciting to see how far you've come, and I can't wait to really get a chance to take a peek into the browser that was shared when the time comes, and to think about some of the initiatives that you guys will be uniquely positioned to be able to take on. And it was great to see the research you've already done.
Yeah, and I guess just again, thanks for inviting me to participate today. Certainly, Peter, to your comments, I think there's oftentimes a pre-competitive space where you can find real engagement across industries, whether it's diagnostics like Illumina, pharmaceuticals. It's finding that pre-competitive space where there's shared alignment that I've seen the most productive collaborations move the quickest.
Last word to you, Alan.
I'll completely agree with what everyone said. And just to borrow the punchline from my last slide as a conclusion, let's get on with it.